Hello everyone, this is a new session at Aloasis Academia, and in this session we will learn more about the methods of studying cells. So in the first video we covered everything there is to know about microscopes, how they are used, the specific terms that you need to understand, and in this video we will cover everything there is to know about cell fractionations, from homogenizations, ultracentrifugation. So let's start. So, cell fractionation. So, cell fractionation is the process where cells are broken up and organelles are separated from each other. So, as we look here in this diagram, we have a cell membrane that is intact. So, what happens is that during cell fractionation, we will break open the cell, and this will result in the organelles start moving out of the cell through these openings, as we can see in this image here. So why do we use cell fractionation? So the main important thing that you need to understand is that cell fractionation is important to studying the structure and function of different organelles inside the cell. Because once we manage to get these organelles moving out of the cell, we can then collect them and then separate them, and then we can then do some research in terms of structural research and functional research as well. Okay, so now we get to the process of cell fractionation. So let's say that you want to conduct an experiment, and in that experiment you want to extract the mitochondria from the cell, and you want to do some experiments on that. So how do we extract the mitochondria from the cell or the tissue we have? So now we will go through the preparation of tissues before cell fractionation. So which means just preparing the tissue before we break open the cell and extract the organelles, especially or specifically the mitochondria that we are interested in. So before breaking the cells, what we need to do is to put the tissue that we have in a special solution. And that solution, it's special because it has specific conditions that they need to, or requirements that they need to meet. Otherwise, the tissue will be ruined and the organelles will be damaged. So the first important thing about this special solution that you put the tissue in is being cold. And why we need the solution to be cold? Because cold temperature reduces enzyme activity, and this prevents enzyme from breaking down the organelles. So if we leave the tissue at room temperature or in room temperature, what happens is that the enzymes will still be active and they will just break down the cell and damage the organelles. And we don't need that because we need to study the mitochondria in an intact way. We need it still be intact and functional. So that's the first thing, is having the solution to be cold. The second thing is the fact that the solution need to have the same water potential as the tissue. And why is that? Because having the same water potential as the tissue will avoid us any damage caused by osmosis. So if, for example, the water potential outside the cell was higher than inside the cell, this will result in the water entering into the cell through osmosis and will burst the cell. Bursting the cells, it means we just lost all the organelles and they are damaged. The other one is if the water potential inside the cell is higher than outside, then it means the water will move outside of the cell through osmosis into the special solution, and this will result in the cells in cell shrinking and damaging the organelles inside. So the second important point is to have same water potential as the tissue. The third point is for the special solution to be buffered. Buffered mean has a stable pH. And why we need a stable pH? Because changes in pH will result in altering the organelle structure and also altering and affecting enzyme function. So if we get like a very low pH, the enzymes might stop function. This might damage the mitochondria, and we don't need that. We need to make the tissue as intact as possible until we extract our organelle of interest. So the buffer keeps the pH constant. So there are very important keywords that you need to remember in this slide here. And once you are writing your answer in the exam, you need to include these specific keywords to gain the marks. Okay, so after covering the special requirements of the cold isotonic solution or the special solution that we preserve the tissue in, then we go to the two stages of cell fractionation. The first stage is homogenization, which is the process during which the cells are broken up using a homogenizer. which is a blender-like device. Homogenizer can be a scalpel, which like these small knives that are used to cut the tissues, or any other type of blades that can cut the cell and allows us to release the organelles. So if you are asked in an exam what happens during homogenization, what happens is that basically cell membrane breaks and organelles are released. The resulting liquid after homogenization is called the homogenate. So homogenate is the resultant liquid. So as we can see in this image here, we would have our special solution with the tissue, so cold isotonic solution, isotonic buffer solution with tissues or cell samples that we want to study and extract, for example, as we said, the mitochondria from. So we need to go through homogenization, where a homogenizer will break the cell membrane and release the organelles. Once we get our homogenate, we need to filter it. Why? Because we need to remove whole cells and large debris. And remember, because the whole process of cell fractionation is that because we need the organelles which are inside the cell. We're not interested in whole cells or large debris. So once the homogenate is being filtered through a funnel, all the large debris and the whole cell remains will just stay in the funnel in the top, and then all the filtrate will, which is called the filtrate. That's what we name the solution after we filter it. That will contain mixture of organelles. Okay, we still hasn't isolated the mitochondria, but we know now that the mitochondria is going to be here among other organelles, and that's the first stage of cell fractionation. So now we get to the second stage of cell fractionation, which is called ultracentrifugation. So what's the process of ultracentrifugation? It's basically that the fragments in the filtered homogenate that we created in the last step, we separate them using a machine called centrifuge, and that's a centrifuge. And the way the centrifuge works is by spinning at very high speed. So centrifuge can be machines like that, can be other machines like the ultracentrifuge, where you have a tube like that. Within that tube, you will have the filtered homogenates, and then the machine will keep spinning at very high speed. So now the question: why centrifuge are spinning and using high speed to separate the organelles? Is because spinning at high speed will result in something called will create something called centrifugal force. So centrifugal force is what separates the organelles within the tube based on two main factors. The first one is based on size or mass of each organelle, and the second one based on the density of the organelle. That means that heavier organelles will move down first. So heavier organelles will be there at the bottom of the tube after spinning or while spinning, and lighter organelles will be at the top of the liquid here. Okay, so the basic idea of ultracentrifugation is that at each increase in the spinning speed, what happens is that the next heaviest organelle moves to the bottom of the tube. And as we said that in ultracentrifugation we will use a tube with the filtered homogenate that we created in the first step. And what happens is that the increase in the spinning will result in the heaviest organelles being precipitated at the bottom of the tube, and then the lighter organelles will be suspended in the liquid here. The keywords that you need to remember that this precipitate, it's called the pellet, and this liquid, it's called the supernatant. So supernatant contains the organelles that are lighter, and the pellet will contain the organelles that are heavier, which which they moved down to the tube during centrifugation. So during the process of separating the organelles, ultracentrifugation will be done through different steps. So the first two steps, they will include two spins at low speed. So what happens is that the homogenate we made in the first step during homogenization we will place it in the centrifuge, which means that just the tube that we have made in the during homogenization with the homogenate, we will just place it in the centrifuge, and then the centrifuge will be spun at low speed. The speed we can start with 100 G, then we increase it to 1000 G. So what does G mean here? It just basically, it means, so for example 100 G, it means it's 100 times the acceleration due to gravity. So that what exactly it means. This one, it just means 1000 times the acceleration due to gravity. And that's referred to the how fast the centrifugation is occurring at. So as we said, the first tube, you would have the suspension of broken cells containing subcellular components. Then we will do the first spin at 100 G, and this spin will result in pelleting the whole cells and all debris. Okay, so all the organelles will be suspended in the supernatant. However, the pellet will have the whole cells and debris. This spin in specific has very similar function to the filtering funnel during the filtration of the homogenate. It's just to remove whole cells and debris that we are not interested in. We are interested more in the organelles there. So what we do with the pellet, we just dispose it in the bin or in the waste, and we do not use it. What we are interested in is the supernatant, because the supernatant will contain a mixture of organelles that were released from the cells. So to start now extracting and separating our organelles one by one, what we do is another spinning, but this time the centrifugation gonna be at 1000 G. And here you need to remember that what happens during centrifugation is that the heaviest organelle move to the bottom first, and the heaviest organelle in the cell is the nuclei, is the nucleus. So since we would have a lot of cells that were broken in the homogenate, we would have the nucleus of many cells, and that's why all the nuclei within the supernatant will be pelleted and precipitated at the bottom when we spin the centrifuge at 1000 G. So we will name the first pellet here because we are interested in that. If you are interested in studying the nucleus, then you will use this pellet here, which we will call it pellet 1. And then we will have supernatant 1, which contains the other mixture of organelles that are lighter than the nucleus. That's why they are still in the supernatant. However, the nucleus is gonna be all the nuclei, they're gonna be pelleted in the precipitated here. So you might get asked, why do nuclei pellet first? And as we said, because they are the largest and densest organelles, and therefore they are experiencing the more centrifugal force. So if the object or the organelle is experiencing more centrifugal force, it means it's larger and denser than other organelles. In the third spin, we will use medium speed in this spin. So what happens is that in the second spin, we created supernatant 1 with all the organelles that are lighter than the nucleus. So what we will do, we will transfer this supernatant into a new tube, and then we will spin that tube at even higher speed, which is at 3500 G. And this centrifuging the tube at this speed, what happens is that the next heaviest organelle will be precipitated and will form the pellet, and in this case it's going to be the mitochondria. So spinning the homogenate at 3500, this will allow us to precipitate the mitochondria, and we get it in the pellet. So why the mitochondria is pelleted at this stage? Because they are lighter than the nucleus. That's why they are not they do not form the pellet in the second spin at 1000 G. However, they are forming this the pellet at the third spin, which is at 3500 G, because they are still heavier than other organelles. So what we have in supernatant 2 here is that any organelles that are heavier, that are lighter than the mitochondria, and then the mitochondria will be pelleted here, and then we can take the mitochondria and do some experiment to study their structure and function. And now we see why cell fractionation is a very important process, because it allows us to reach to this point where we have, if we are interested in studying mitochondria, we will be able to separate the mitochondria from the rest of the organelles and do some independent studies on it. And now we get to the fourth spin, where we spin the tube at a very high speed. So what happens is that now we will collect supernatant 2 that was produced in the third spin. We transfer it to a new tube, as we can see here, and then we spin this tube at even higher speed than the third spin. We will spin it here at 16,500 G. Okay, spinning the homogenate at this high speed will allow us to get a pellet with lysosomes that has lysosomes. And that's why it's important to spin at this speed, because the lysosomes are small organelles, so they need really high spinning speed to get enough strong centrifugal force to pellet the lysosomes. And this will form pellet 3 and supernatant 3. So any further centrifugation after that will allow you to pellet other organelles based again on the size or mass and the density of the organelles. So the next heaviest organelle after lysosomes is going to be endoplasmic reticulum. And then even if you spin the centrifuge at even higher speed, you will get the ribosomes. Okay, so now this takes us to like a recap slide to understand and to remember why ultracentrifugation is important. So first of all, it allows scientists to separate organelles, study each organelle individually, and understand their structure and function. And that's very important, because if scientists wanted to study any disease that is related to a specific malfunction of an organelle, then they would need to separate the organelle and do some research on it. And that's what ultracentrifugation allowed scientists to do. That's why ultracentrifugation led to major advances in biological knowledge, because scientists could finally see what each organelle does, and they could separate each organelle individually. Okay, key take-home points. First of all, cell fractionation is used to separate organelles, so scientists can study the function and the structure of organelles. This involves two main steps: homogenization, which breaks the cell and open them; ultracentrifugation, which separates organelles based on the mass and the density of each organelle. During centrifugation, organelles separated from the heaviest to the lightest. So the heaviest is the nucleus or nuclei, then mitochondria, lysosomes, endoplasmic reticulum, and the lightest is ribosomes. To get a full revision, you need to have a look at our revision booklets, which are found on my online store, Payhib. So in this booklet, you will have a full keywords bank for this topic in specific. So you're going to ace your exam, because you will find all the keywords that are the examiners looking for to gain the marks. Please don't forget to like, comment, subscribe, and turn on the notification button, so hopefully you will be notified of our next video. Thank you.